All right, let's step into the role of medical detectives for a bit. We're about to tackle a genuine paradox, a condition that, get this, causes dangerous blood clots inside the body, but at the same time makes a key lab test for clotting run slower. How is that even possible? Well, let's dive in. So here it is. This is the central puzzle. It seems completely backward, right? You've got a situation that promotes clotting, but in the lab, it looks like the exact opposite. This is the very question that baffled scientists for years, and trust me, the answer is absolutely fascinating. So, who's the main suspect in our mystery? It's a condition called antiphospholipid syndrome, or just APS for short. Let's get to know it a little better. Okay, so at its heart, APS is an autoimmune disorder. You know, that's where the body's immune system gets confused and starts attacking its own healthy tissues. In this specific case, it makes these rogue antibodies that go after phospholipids, which are super important parts of our cell membranes. And that attack is what kicks off the formation of blood clots. Sometimes APS just shows up all on its own, we call that primary APS, or it can tag along with another autoimmune disease, most famously with lupus. Now, to diagnose something this complex, doctors can't just guess. They need a really clear set of rules for gathering the evidence. For APS, that framework is called the Sapporo criteria. What I love about these criteria is how they split the diagnosis into two simple parts. On one side, you have the clinical criteria, basically what actually happened to the patient. We're looking for proof of a major blood clot or some very specific and serious pregnancy complications. And on the other side, you've got the laboratory criteria. What are we finding in their blood? This means we have to find at least one of those key antibodies. But, and this is crucial, we have to prove it's not just a fluke. The test has to still be positive at least 12 weeks later. So, in the lab, we're on the hunt for three main culprits. You can think of them as a team, and each one has a special job. First up is the lupus anticoagulant, or LAC. This one is the most specific. If you find it, it's a huge pointer directly to APS. Then you have the anticardiolipin antibody. This one's the most sensitive. It's the one that's most likely to pop up on a test. And finally, there's the anti-beta-2 glycoprotein I antibody. A lot of people consider this one the most important because it's the real engine behind the actual clot formation. Okay, so we have our key player, that lupus anticoagulant antibody. Now it's finally time to crack this case wide open and solve the mystery of that slow clotting test. Let's just put the question out there again, front and center. APS causes clots. It's a pro-clotting state. So why on earth does this lupus anticoagulant antibody make the APT test, which measures how long it takes blood to clot, run longer? It just doesn't make sense. Well, it's time for the big reveal. And here is the absolutely brilliant solution. It all comes down to the environment. Inside the body, or in vivo, as scientists say, the antibody attacks and injures the cells lining our blood vessels. That injury is the trigger that sounds the alarm and kicks off the whole clotting process. But in a test tube, in vitro, there are no blood vessel cells to attack. So the antibody, with nothing else to do, binds directly to the clotting factors and phospholipids that are needed for the lab test to work. It basically grabs them and takes them out of the game, which slows the whole reaction down. It's the same antibody, but in two totally different environments, you get two totally opposite results. Mystery solved. Okay, but how can the lab techs be absolutely sure that the long test time is because of this sneaky antibody and not because the patient is, just missing some clotting factors? Well, there's a really clever test for that called a mixing study. Here's how this little piece of detective work goes down. First, you take the patient's plasma and mix it with normal plasma, which is full of all the clotting factors you need. Now, if the test time becomes normal, well, problem solved. The patient was just missing a factor. But if the test still runs slow, that tells you an inhibitor is in there, our lupus anticoagulant antibody. And it's so powerful, it's messing with the healthy factors too. And here is the final brilliant move. You add a bunch of extra phospholipids into the mix. The antibody gets distracted and prefers to bind to all these new phospholipids, which means it lets go of the clotting factors. The factors are now free to do their job, the test time corrects, and boom, you've just confirmed the diagnosis. Now that we've solved that fascinating lab paradox, let's bring our focus back to where it really matters. The real and often life-threatening danger of antiphospholipid syndrome inside the human body. So how exactly do these clots form? Remember that most important antibody, the anti-beta-2 glycoprotein I? This is where it does all its damage. See, normally, when platelets get old and are ready to be retired, they put a little "remove me" sign on their surface. A protein comes along and binds to that sign, marking the platelet for disposal. But in APS, this antibody attacks that whole complex. So instead of the old platelet being cleared away quietly, the attack causes it to get sticky and clump together with other platelets, and that's how you get the dangerous clots that define this disease. And to really understand just how serious this condition can be, we have to talk about its most severe form, catastrophic APS. Now, it's rare, thank goodness, but it is a devastating complication. It's when widespread clotting happens incredibly fast, damaging 3 or more different organ systems. We're talking kidneys, lungs, the brain, all in less than a week. It is a true, life-threatening medical emergency. So, we've managed to solve one pretty incredible medical mystery today, seeing how the very same thing can cause one problem in the body and the complete opposite problem in a test tube. It really makes you wonder, doesn't it? What other conditions are out there, hiding a similar kind of paradox, just waiting for us to figure them out?